Okay, brilliant. Thank you, Roberto, for the kind invitation to speak today. So, yeah, my name's Chris Stape, and I'm going to be talking to you today about some work we've been doing on single-cell phenoscaping of tumor microenvironment organoids. So, my lab's interested in cell phenotypes, so not just what cell phenotypes exist, but also what controls cell phenotypes. So there's lots of different things that can control cells, including things like cell-intrinsic features, so these are epigenetic states, transcriptional networks, mutations, but also things that come from outside the cell, so extrinsic factors such as growth factors, mechanics, metabolites. And in cell biology, we're often looking at one or two of these things in isolation. So we're looking at some of these things in isolation, but actually they're often done together. So what my lab's really interested in is how we can functionally map thousands of phenotypic regulators. So I'm going to stand over here. So to do this, my lab's interested in this concept of phenoscaping. So this is a functional map of phenotypic space. And this is a bit of a buzzword at the moment. It's something that people are kind of interested in single-cell technologies, and it's something that we've been developing a lot in my lab as a way to study cell-cell communication and tumor microenvironment. So we're interested a lot in single cells, and you're used to seeing one dot equals one cell kind of plot. So every time you see a t-SNE or a UMAP, you'll be looking at cells in this kind of format. But what I'm inviting you to think about today is to think about one dot as a single condition, because as these data sets grow and we start looking at lots of different conditions, we can start exploring phenotypic space. So these conditions, so these single cell data sets that we collect, they exist in this large phenotypic area. And to traverse this area, to explore this area, we need to perturb the system. We can't just look at static snapshots. So a single perturbation, so for example, a growth factor might make the system more STEMmy, push it towards a more stem cell-like fate, and you can explore this phenotypic space. Then a different perturbation may lead to a different state. So for example, the cells might start quiescing or dying or going to a different state. So you've got this phenotypic space, and phenotypic space is finite. It's not an infinite thing. A cell can't just become anything. And the way we can explore all the possible states cells can operate in is by just perturbing them and seeing what happens. So it's classical cell biology. But the idea here is we're doing this with single cell resolution. So in a phenoscape, what we're doing is just taking this concept and just scaling it up, basically, and just doing thousands and thousands of perturbations. So these can be growth factors, mutations, microenvironmental cues, matrix stiffnesses, all sorts of things. And the crucial thing to observe here now is when you look at these kind of embeddings, each dot is a single condition. It's not a single cell. But inside each dot are thousands of single cells. So you will see embeddings in my talk today that look a bit like single cell plots, but actually each dot is a condition. And if any of you have ever done omics analysis and then on a PCA, for example, of that afterwards, and you see your dots moving around in a PCA plot, that's essentially what you're doing. But in the PCA example there, each dot comprises a gene list, but for us, each dot now comprises a cell list with features per cell. So how do you do this? It's a sort of conceptual idea of mapping out phenotypic space. How do you build a phenoscape? Well, first of all, you need a very dynamic model system. Now, I work on solid tumor biology. I work on colorectal cancer. So for us, we use organoids, but in principle, there could be cell models that would also work for this. It needs to be high throughput, and it needs to be dynamic. It needs to be able to change and biomimetically model the biology that you're interested in. We also need thousands of perturbations, so you can't just look at one or two things. You need to be able to do an arrayed perturbation screen. You need to be able to look at different things like genetics, ligands, drugs, stromal cells, and it needs to be arrayed. A lot of single-cell screening, especially in the genomics area, is using pooled screens, and that's great if you've only got intrinsic factors, but if you want to see what's happening in a microenvironment, you can't do that. You need an arrayed format. And then, of course, you need a way to quantify single-cell phenotypic data. So normally at this point in the talk, people expect me to reveal some sort of single-cell RNA-seq technology that will do this, but obviously, with the context we're in at the moment, you know what the answer might be here, and for us, it's cytof. But it's a particular type of cytof. So my lab developed a few years ago a technology we call thiol organoid barcoding in situ mass cytometry, so TOBIS mass cytometry. And it's basically a bit of a hack on traditional cytometry workflows, because we're not dealing with PBMCs or a nice sort of suspension of cells. We've got cells grown in matrigel in a 3D environment with lots of sort of mess and complexity that comes with that. So what we do is we have a multivariate organoid experiments. This can have drugs, co-cultures, or CRISPR reagents, for example. And then what we do is we fix all of the cells in situ, so we fix them while they're inside a 3D matrix in PFA. We do an IDU pulse as well and some phosphatase inhibitors, and then we barcode them using these Tobis reagents. So the Tobis reagents are based on TMAL, tellurium malamides, which I think are now commercially available. At the time, they were part of a collaboration we did with Mark Nitz in Toronto. But they're basically thal-reactive probes with the tellurium, a monoisotopic tellurium on the back end, and we also pair it with some cisplatins. Now, the reason we use a thal-reactive probe is because amine-reactive probes just don't work when you've got loads of matrigel. So if you're dealing with lots and lots of ECM, you can't stain organoids because all the amine-reactive probes just bind to the ECM itself. Whereas the thal-reactive probes, they'll go straight through matrigel and they'll bind just to cell surface ectodomains. So this is because ECM proteins basically don't have any disulfide bonds in them, whereas cell surface proteins have lots of oxidization and reduction happening in the ectodomains of their proteins. So you could easily probe cells in a deep, sort of thick, protein-rich ECM with a thal-reactive probe. So we use this thal-reactive moiety on the end with a monoisotopic backbone to build a barcoding system, which is kind of inspired by the work of Vili Zunder and others with the original barcoding system that's used in cytography. We use 126-plex barcoding, so I normally have to explain this a lot to people, but this is a cytoph audience, so you understand this. We use a 9-choose-4 setup, so we've got our first barcode here, red is on and black is off, so 4 on and 5 off, going all the way through the system until 126 at the back end. Now, these are all organoids that have been stained in situ, so all this barcoding is happening inside the plate itself. It's not happening in a suspension, which means one person has essentially just got 2 plates to cover the 126-plex, and it's a very easy thing to do with a multi-channel pipette. We then have a little robot that harvests the organoids, or a PhD student if the robot's broken. And then we pull the whole sample very early on. So we kind of have the sample handling benefits of a pooled system, but we use the barcoding it allows us to get an arrayed system into a pooled format quite quickly by barcoding quite early in the process. We then do extracellular and intracellular antibodies. We have a very large intracellular panel because we're largely interested in signaling, and a lot of phenotypes developed, especially in solid tumor biology, are really at the signaling level. So we have probably about 80% of our panel is intracellular. And then we go for the cytoph, you all know this, single cell events, debarcode, and data analysis. So generally speaking, our assays, we have about 30 PTMs, around 6 cell states. So this is G0 apoptosis cell cycle, for example. And then we also have as many cell types we can get good antibodies for, and then 126 cultures. So this allows us to do very high throughput single cell organoid phenotyping experiments in a routine way. So we get millions of cells, all of these different parameters across lots of different cultures in a single run. So it's just the sort of thing you couldn't do with a single cell RNA-seq approach. So we use that to approach these kind of array-style experiments. So here we've got, and you'll see sort of a few cubes in this presentation. This is how we tend to design our experiments. So here we were interested in colorectal cancer mutations. So we've got different mutations here, so APC, KRAS, P53. Then we've got different extracellular ligands. So we've got Wnt, EGF, noggin, R-spondin, lots of different combinations of those. And we wanted to understand how they interact together, but also with epithelial on their own or with stromal cells such as fibroblasts. So it's a big kind of interaction matrix of all these different things. We can then barcode everything, run it in triplicate. So we've got a triplicate system. And then we've got 570 single-cell data sets that come out of this one experiment. And that allows you to chart these kind of this is a proper phenoscape figure. So here 1 dot equals 1 condition, as I alluded to before, and inside each dot is a single cell embedding of different cell types. So here, the question we were interested in is how these different growth factors and oncogenes play together. So here we've got our wild type baseline cells. When we have a wild type or an APC mutation with no Wnt, we kind of get a slight shift upwards in the system. KRAS plus Wnt goes here. KRAS minus Wnt kind of moves to this different state. And then when we have KRAS and APC, which are the 2 major driving mutations in colorectal cancer, or in R-spondin, which phenocopies the same signaling pathway, we get to this what's called a proCSC state, so it's proliferative colonic stem cell, so kind of like a classic cancer stem cell. And we know what's happening with signaling here because we're measuring lots of PTM, so it's high EGF, MAP kinase, PI3 kinase signaling, with lots of beta-catenin Wnt signaling. Conversely, we see when we have wild-type cells with an AP, or just a single mutation, and they get exposed to Wnt3A or also TGF-beta, we get this very slow cycling revival stem cell state. So we can basically use this approach by doing these very systematic perturbation studies to kind of map out the phenotypic space that the cells occupy in this system. We can then partner that with single-cell RNA-seq data. So this is a single-cell RNA-seq Waddington-like landscape plot of all the stem cells at the top. They gradually differentiate down this little valley, and they go into these differentiated cells down here. So this is just the wild-type colon, the healthy setting. If you then have some fibroblasts in the system, so this is a stromal load, so things coming from outside the cell, we get access to this revival stem cell population, which is involved in wound healing and some other things in cancer I'll talk about in a minute. But then in cancer, you get a complete reprogramming of the landscape. So you get basically all the cells become stem cells and get trapped at the top of the Waddington landscape. And this is actually what we call a tarn or a lock, appropriately for Scotland, where you get cells that essentially get trapped at the very, very top of stemness in the landscape. So you can get this from single-cell RNA-seq data, and that's fine. But really, what you don't get from this is how you got between the different states. So by doing all the phenoscaping work that we did earlier, we can assign a mechanistic understanding to how you get to these different states. We know exactly what conditions of signaling need to be on. For example, TGF-beta going into YAP and revival stem cells here. Balanced signaling gives you this. And then when you have mutations and the absence of exogenous ligands, you get into this proCSC state. So by combining single-cell RNA-seq, which gives you this nice overview of cell states, with something like a highly systematic perturbation study with CyTOF, you can kind of get to both these landscapes, but also this mechanistic understanding. So we're also interested in drug interactions, and here in colorectal cancer, we also work with patient-derived organoids, PDOs. So these are coming from patients in the clinic, and they're modeling individual tumor biology. And it's been known for a while that cancer-associated fibroblasts, CAFs, they can affect drug responses in patients. And the more CAFs a patient has in their tumor, the worse they do. So it's something that we wanted to try and understand. It's sort of counterintuitive. You think the more cancer a patient has in their cancer, the worse it would be, but actually the more healthy stromal fibroblasts, these CAFs that are in a tumor, the worse the patient does. So we wanted to try and understand that and how it interacts with drugs. So here we've got another one of these cubes, these arrays. Here we took 10 different patient organoids, so just representing a little bit of patient heterogeneity that you might see in the clinic. And then we treated with lots of different therapies, so chemotherapies, targeted therapies, some preclinical therapies of interest. But crucially, we did it either with PDOs on their own, CAFs on their own as control, or both, because we wanted to understand how the CAFs influenced it. So that's the cube. This is Maria, who did the experiment. So this is all our organoids. So for a normal screening project, this wouldn't be considered that crazy, but to do single-cell analysis on all of these cells at 96-well plate format was quite a lot of work, and nearly broke our Helios a few times. And it's she's very happy with herself in this. She's got this thing on her face here, this smile, which is quite rare for her, but she got the smiles on there because she thinks she's finished in this moment. You know, she spent years doing this, and she's kind of through COVID and everything. But so we realised actually there's a huge problem here because the data was just insane. So we had cell type specific markers, cell state, DNA damage, PTMs, and we had this around 3,500 single cell data sets that came out of this. It's sort of in a big embedding. Now, I don't have time today to go into all the data analysis and exactly how we broke this down, but UCL, where I'm based, and Yale University in the States have an exchange program. So Maria was fortunate enough to be a part of that exchange program. She went to work with Sumita Krishnaswamy's lab at Yale to come up with a new computational method to help deal with this. It's a method called Trellis, and it's published now, but it's an interesting way to study a large-scale cytometry data. So I'd advise if you are working at scale with barcoding or large samples, it's an interesting method. So what Trellis allows you to do is to, again, create these kind of phenoscapes where each dot is a single condition. Here, it's coloured, this embedding here is coloured all by the samples. So here the controls are all on one side, and then gradually as a treatment effect occurs or a therapeutic effect occurs, you spread right across the embedding. Now, the route that you take through this, these dots take through this embedding represents how they responded. So if you get more apoptosis, for example, more DNA damage or more of a particular signaling pathway, you get a slightly different route through this tree. So what we saw when we did this with the drugs is that actually the drugs didn't really cluster by much. We thought, okay, the drugs are doing the same thing on all cells, they'll cluster together. But actually we didn't really see that. What we saw is that they cluster largely by patients. And if there are any, you show this to any clinicians and they're like, yeah, of course, that's what we see in the clinic. Some patients respond really well and some don't, and we don't know why. And that was what we were mimicking with the patient-derived organoids. So we had a couple of groups of patients that had very high chemosensitivity, some had high chemosensitivity with huge DNA damage responses, and then some that were really refractory that just didn't do anything. Then a couple that were a bit more anecdotal, they're very sensitive to just one type of therapy. But the reason we did all this really was more to do with the microenvironment. So here it's coloured by either the PDOs on their own or in green, or PDOs with the CAFs in pink. And you can see some examples here of what we call CAF protection. So for example, there's PDO75 here on the top. It's been treated with SN38 chemotherapy, has a huge therapeutic response, it goes into apoptosis, colonical cell death kind of approach. But then if you take that same organoid with the same drug, the same concentration at the same time, but you put it in with a CAF, it doesn't die. You get this protection effect. And this was something we were seeing routinely in some elements of the data set. So we wanted to understand a bit about why this was happening, and because we're doing this, we're essentially doing a drug screen with CyTOF, we can get mechanistic insight immediately. So it's not just a live dead score that you might get with like a cell titer glow style drug screen. So here each column is an individual condition, and I'm just focusing in here on a chemosensitive and a chemorefractory organoid. And you can see in this blue, so this is the SN38 kind of classical chemotherapy treatment. In our chemosensitive, you're getting S phase and M phase blocking, so you get cell cycle blockage, which is what should happen. You get DNA damage being triggered, and you're getting an apoptotic response. But in the chemorefractories, you also get the on-target effects. You get huge amounts of DNA damage, you get the cell cycle blocked. So these drugs are working, but for some reason they're not entering apoptosis, and we don't really, we still don't quite understand why this is, but it's definitely not because the drugs aren't hitting the target. So being able to use CyTOF in a way to do drug screening is a nice way to get on-target effect in your screen. So you're not just wondering, okay, did it get into the cell in the first place, or did it just bounce off? So we see on-target drug effects in most cases, but we only see apoptosis in a few. So the apoptosis from drug responses is clearly very patient-specific. So we wanted to try and understand a bit about why it was so patient-specific. So we went and looked at the organoid signaling states. So this is nothing to do with genotype. This is nothing to do with where they came from, the stage of the patient, or anything like that. Literally, what is the signaling state of the organoids before they see drug? And we found that they clustered quite discretely, where you've got this very highly mitotic and proliferative population, and then this sort of more slower cycling population to the side. And if you look at the chemotherapy responses in these two groups, it's night and day different. Basically, if you've got a high mitotic signaling state at the point when you see these drugs, you die very quickly. But if you're quite slow cycling, if you're quite a relaxed cancer cell in many ways, you're really chemorefractory. And you couldn't discern this from genetic information. This is something that we can only see at the signaling level, at the protein level. Now, really, we did all this because we were interested in CAFs and how the stromal microenvironment works, so this kind of intrinsic versus extrinsic idea. And so, again, because we've got PTM information in our data set, we can actually look at the signaling pathways during the drug screen itself. So this is PDO21 on its own, and it has this kind of little flowery shape. And then when it goes in co-culture with CAFs, it kind of balloons into this big like cyst-like thing. And that's accompanied with a big signaling change, largely with PI3 kinase and MAP kinase shifts that we see, an increase in inflammatory signaling like NF-kappa beta. Conversely, with a PDO that doesn't receive this, so for example PDO27, it just looks the same in both cultures, and the signaling, if anything, it just generally goes up a little bit, but you don't get this switch-like response, and you certainly don't get this morphology response. So we know that YAP signaling is very important in these organoids. So we went in to look at nuclear translocation of YAP. We see that again also in the co-culture environment. So YAP's quite hard to detect with CyTOF because it's a protein translocation. It's not really a PTM-driven response, but we could still see that here. So that kind of gave us some reassurances that we had this general PI3 kinase low, MAP kinase low, YAP high, which looked very similar to this revival stem cell signature that we saw in the first part of the talk. It was a very similar signaling profile. So we wanted to confirm that, so we went back to single-cell RNA-seq. So this is sort of an example where we use CyTOF and single-cell RNA-seq in partnership. Each dot here is a single cell, and it's spread across a treatment effect using MELD likelihood. We've got a proCSC signature here, so this is the gene signature for the proliferative colonic stem cells. PDOs in green, CAFs in red, in replicates. And generally speaking, the PDOs, when they're on their own, they're very proliferative, classical cancer stem cells. But when they go into co-culture, you lose this signature, and that can be quantified here. And then conversely, as they lose this signature, they gain a new one. They gain this revival stem cell profile. And we could then go in and look at the gene signatures and also see that there was a YAP effect, just as we thought there would be from the IF earlier. So we kind of start getting a sense of there's this plasticity of the system, this switch that's happening between these very proliferative cancer stem cells and these revival stem cells that we saw earlier. And these revival stem cells are normally induced, they're not normally found in healthy biology, they're found in tissue damage and responses to infection. And we're now seeing this in cancer as well, that this is a major feature of tumor biology, this plasticity. Now, because we understand a bit about the mechanism, because we're doing more of a mechanistic focused drug screen in this whole phenoscaping approach, we can now take this this knowledge and undo it. So this is the organoid on its own, put it with the calf, turns into this cyst. And then if we put verteporfin in, which is a very kind of basic, trivial YAP inhibitor, YAP translocation is avoided, and then the organoids don't turn into this shape. So by understanding a bit of the mechanism, we can kind of start controlling the system. And then, again, using cytoff, we can do apoptosis analysis of this. So this is PDO apoptosis. When it sees the chemotherapy drug, you get a treatment effect. When you have the CAFs in there, they go into this cyst and they get protected. But if we put the YAP inhibitor into the system, we can resensitize. So again, understanding mechanism during a screening process, I think, allows you to shortcut into rational resensitization of these therapies. So we now sort of see these revival stem cells that we've identified using this cytoff approach as these plastic drug-tolerant persister cells in colorectal cancer. And this is actually now sort of a major clinical target. So to go back to our Waddington plots, you can see here, so one dot is one cell here. You can see all this, the proliferative colonic stem cells sort of sit at the top. They're just sitting there being stem cells in cancer. Tiny little bits of differentiation, but barely anything, because they're kind of really trapped in this stem-like state. And they're very proliferative. So this is colored by Chi67, and they're really kind of very proliferative. And we know that they have this particular signaling signature. But then what happens when the fibroblasts turn up, so these extrinsic factors start coming in, it allows them to explore more phenotypic space. So we've got here these revival stem cells, and they're very slow cycling, and they're very pro-survival. They're very robust cells that can withstand treatment effects both from chemotherapy, and we're now finding from radiotherapy as well. So, and this transition happens in we think about 2 hours. So it's a signaling shift. It's not some big acquired mutational event. It's very much a signaling protein level switch, and probably a few epigenetic factors playing in as well. So all of this has led us really to a new model of plasticity in colorectal cancer, which excites me a lot. I appreciate you're probably not colorectal people, but I think that the takeaway message here is that we now know that cell states and cell phenotypes can change so quickly, and they can have such important impacts on cancer that we can't see these things using genomics. You know, if you were to sequence all these cells that I've just showed you today, you barely would see any differences. But when you start looking at the protein level and you start looking at function and PTMs, you can see it very clearly. So we have these very proliferative colonic stem cells, the revival ones that I've talked to you about. We've got this transition between them. And we now know from the phenoscaping, from systematically going through and looking at all the different conditions, that oncogenic mutations generally push you towards this direction. I should say this one in the middle is kind of what we've all got hopefully in our guts at the moment. It's a normal kind of balanced colonic stem cell. But then if you have too much stromal load, so too much extrinsic factors, whether it's ECM or inflammatory signaling like prostaglandins, TGF-betas, WNTs, things like that, it pushes the cells towards this survival state. Now, in homeostatic biology, this is fine. This could be involved in a wound healing response. But if this happens in cancer, these cells are essentially protected from therapy, and they cause residual disease. We can kill these really easily. Chemotherapy kills these quickly. Radiotherapy kills these quickly. But as soon as a cancer cell starts becoming slow cycling, becomes a bit more differentiated, becomes a little bit less like a cancer cell, it's really hard to kill. So understanding these transitions is sort of fundamental really to what we're doing in cancer. So yeah, that kind of brings us back to this extrinsic and intrinsic balance that I kind of started with at the beginning. By studying both at the same time in a systematic way, we're starting to unpick a bit about what's happening in these systems. And again, I know we're here to talk a bit about cytoff today, but this could not have been done any other way. We couldn't have done this with single-cell RNA-seq. I don't think even a lot of high-content microscopy approaches could have approached this, because you need so many markers per cell in order to resolve these features. So yeah, thanks to SPT for making the cytoff, because otherwise we couldn't have done this. So there, I'll finish and thank the people who did the work. Most of what I showed you today was from Zhao, Maria, and Farham, and Jahangir, with lots of organoid help from Petra. Really indebted to Mark and Jamie at Toronto, who developed the T-mail reagents, which I think are now commercially available, or at least on a beta program. They're fantastic reagents. I think the batch that SPT is selling is actually the batch we made as a part of developing this. They're great reagents, so I definitely recommend trying them if you're interested in expanding your barcoding opportunities. And of course, Smita and Alex at Yale who helped us with the data analysis. So with that, I'll stop. Thank you for your attention, and I'd be happy to take any questions.